I'm Donna today because Donna couldn't be here. And I asked Donna to send me an introduction to Liz, and she sent me several paragraphs that were effusive and lovely and would take 5-10 minutes to read. So I'm gonna skip over those and just say that Liz is amazing. And that and I'll send her what Donna said so she knows what Donna, because it's very nice. But that she did her bachelor's degree in health and nutrition science at City University in New York, and then worked at Columbia in a lab, and has 14 publications before she even came to us here in Madison. So she's just off and running, and then is doing really cool things with dissecting mice. And we're gonna all get through this together. So
Thank you.
Okay.
Hey, everyone. Hi. So thank you, Jake. That's really nice. Today I will be talking about the impact of maternal immune activation and sex-differential biology on brain function and development, which I think it's really cool because they can focus a lot in bacterial, and I am going to be focusing on viral infection for my projects. Just to start us off with a little bit of the lab, because it makes sense. I wanted to introduce some of the work that's done in the lab by the members. So Joe actually uses huge human data to study why same genetic variations yield different outcomes in people, and he uses neurodevelopmental disorders as his model system. Then we have Kyle, who also uses human data, but now is moving into mice models to study how environmental chemicals can affect neurodevelopment. We also have Roy, who just joined us, and Roy is really interested in understanding how sex differentiation and biochemical pathways affect neurodevelopment. Then we have Kevin, who is our research specialist. It's the person that basically creates most of the data, keep the mice alive, and make sure the Well Lab is not falling apart. And then there's me, and I focus on understanding how a mom getting sick during pregnancy can affect the brain development of the fetus during pregnancy, but also later on in pregnancy. And when you put all of this together, the lab really focuses on understanding how genetic variation and sex-differential biology influence the risk of neurodevelopmental and neuropsychiatric condition, specifically that of autism spectrum disorder. So what is autism? The National Institute of Mental Health describes autism as a neurological and developmental disorder that affects how people interact with others, communicate, learn, and behave. And although autism can be diagnosed at any age, its symptoms as described as developmental disorder because symptoms generally appear at the first 2 years of life. Now, when you think of autism spectrum disorder, you should be thinking of it as a linear disorder, because it can affect a lot of stuff. So it can affect emotional regulation, interest, speech, anxiety, social differences, and even eye contact. And it affects everyone that has autism at a different level. Each one of this is affected at a different level. So when you think about it, you should be thinking more of a variable wheel, if that makes sense. So. Another really interesting thing about autism is that it affects males at a higher rate than females. Right now, the ratio is 4:1. So per each female that gets diagnosed with autism, 4 males get diagnosed. But this sex disparity is something that you observe a lot in neuropsychiatric, neurodegenerative, and neurodevelopmental disorders. For example, autism, schizophrenia, ADHD, and learning disabilities affect males at a higher rate, whereas Alzheimer's, depression, and bipolar disorders affect females at a higher rate. So what is causing this? Well, this sex disparity is stays the same through the years and through states. So actually, yes, okay. So this data, this graph that you see here, it's something I have put together using the CDC data on autism diagnosis from 2017 all the way to 2020, which is the most recent data they have. The blue bars represent males and the red represent females. And this is the prevalence per 1,000 children. So as you can see, through the years, that sex disparity stays the same. It really doesn't change. And when you look at the states overall, over places in the states, it also stays the same. So this is actually data from 2007, and this is looking at the age adults between 18 and 84 that have been diagnosed with autism. So now the question is, what does maternal immune activation has to do with all of this? Why is it important? So I'm just gonna click. So what we believe is happening is that during a viral infection during gestation, this activates a mom's immune system. And this leads to a lot of cascades that are being created by the immune system. And the molecules created by this cascade are actually crossing the placenta and reaching the fetal brain. And then this leads to long-term postnatal effects in health, neurodevelopment, ability, communication, behavior, and social skills. Now, this is not the first time this connection has been made. This is a graph I take out of Python because it took a lot of reading to make it. So the initial hypothesis was actually made in the late 1990s, early 2000. And it was Dr. Machon's study suggested that there's a potential link between prenatal exposure to infectious diseases and the increase of schizophrenia. So that was the hypothesis. Later on in 2003, Dr. Xi actually came out and said, well, it is not what's affecting the baby. It's not the infection itself, but the molecules that mom's immune system is creating. Later on in 2007, which is where I like to think and believe this is where everyone started studying this, was because Dr. Patterson was able to make the connection using mice that show that if a mom gets pregnant during the second trimester of, if a mom gets sick during the second trimester of pregnancy, it can lead to autism-like disorders in mice. Then in 2008, Dr. Fatemi found that genes that are associated with schizophrenia and autism actually get altered if a mom's immune system is activated during pregnancy. Between 2012 and 2015, human studies, and there was human studies that now associated getting sick during pregnancy to schizophrenia. So now there's proof with humans that this is actually happening, but it's with schizophrenia. Then in 2017, Dutra and other epidemiological studies found out that autism risk increased during mid-pregnancy infection. Then in 2018, sex-differential effects were associated to stress response, so the response to having an infection during pregnancy. Now that this is all known, you're probably wondering, so what do you want to do? Why is this important? Well, something that we don't know is the sex differential effects of MIA if they're coming from the gonadal sex or the chromosomal sex. And this is what I'm trying to understand. So what mechanisms are responsible for this sex disparity? The way I want to answer this question is by two aims. So the first one is identifying offspring's behavior impacted by maternal immune activation and chromosomal and hormonal sex. And our expectation is that the offsprings of mothers who had their immune system activated are going to show more anxiety-like behavior and less social recognition. And this is going to affect males at a higher rate. For aim 2, I want to quantify the transcriptomic effects of maternal immune activation and chromosomal and hormonal sex in offspring. And again, we believe that these changes of genes are going to be involved in the function of microglia and astrocytes. And this is just a little breakdown. So aim 1 should help us test for the behavior of XX, XY versus testes and ovaries. Aim 2 should help us look a little bit deeper into the regions, the cell types, and the genes that are affecting these mechanisms. So how am I going to be able to study chromosomes away from hormonal effects? The way I have proposed is by using the 4-core genotype mouse model, which is a mouse model that allows for experimental decoupling of sex chromosome and gonadal effects. So the four-core genotype mouse model is actually a transgenic mice, where they took the SRY gene, which is the gene that decides if you're getting testes or not, and they took it out of the Y chromosome and introduced it as a trans gene in an autosome. So when you cross this animal with like a wild-type female, instead of having your phenotypical XX with ovaries and XY with ovaries, you can actually get XX with ovaries, XY with ovaries, XX with testes, and XY with testes. And this is going to help us see the differences due to sex chromosomes and differences due to gonadal sex hormones. So the project, it's broken down into two stages. The stage one help us answer how does MIA affect wild-type male and female behavior and brain gene expression. Think about stage one. Stage one for us is more like a pilot test. We want to make sure that the things we're going to be running later on actually work. So that's why we did it. And this is a little scheme. So the mating is just wild-type male and female mice, and then embryonic day 12.5. We do an IP injection of poly-IC. And there's two ways you can trigger a maternal immune activation in mice. Well, there's more, but the ones that are widely used are poly-IC and LPS. So LPS mimics a bacterial infection, which is not what I'm interested in. And then poly-IC mimics a viral infection. So that's what we are introducing to our mice. So that leaves us with two treatment animals, so poly-IC and control animals. Our control animals are injected with saline just because we want to make sure that both mothers are getting the same stressors during pregnancy. So we are taking that into consideration. And then at postnatal day 3, I start behavioral testing. After behavioral testing is done at postnatal day 80, we do brain dissection and genomic analysis. For stage 2, which is where we introduce the four-core genotype mouse model, it stays pretty similar, with the differences that instead of having a wild-type male, I'm crossing with a four-core male. We do the IP injection at the same stage, embryonic day 12.5. We do treatments, and then at postnatal day 2, we collect tissue to genotype the mice. And the reason why we have to do this is because when you wean mice at postnatal day 18, you can tell their sex by looking at their external reproductive area. However, with the four-core genotype mouse model, we're not going to be able to do that because their external reproductive area is not going to match for 50% of them. It's not going to match their internal reproductive area. So we have to do genotyping early on before we move on to behavioral assays. And then again, postnatal day 3 behavioral assays. When they're done, we do brain dissections and then genomic analysis. Now, the behavioral assays we did for stage 2. So at postnatal day 3, 6, 9, and 12, I did what we call publicization, and this is helping us test for their communication skills. After they were done at postnatal day 12, we let the mice rest, develop, become basically adults, because we want them to have that hormonal stage active to keep going through behavioral assays. So after postnatal day 16 is what I call like the real behavioral assays. And we start with olfactory preference. Then all the mice get a day rest between assays, so they are not super stressed. So olfactory, then we move to open field, elevated plus maze, 3-chamber, and through social interaction, which help us test for social interaction, reproductive interaction, and resident intruder behavior. Now, why are we doing this test? What is the purpose and the rationale for us to do this? So for publicization, we want to test for the communication behavior, and this is an autism trait. For olfactory preference, we want to make sure that the olfactory function, smell, and discrimination works. So this helps us control for olfaction, and so it is also a sensory comorbidity of autism. For open field, we're testing for motor behavior and anxiety, and this is an autism comorbidity. For elevated plus maze, we're testing for anxiety, and it's an autism comorbidity. 3-chamber, social recognition, and it's an autism trait. Through social interaction, predicting social interaction, and it's autism trait. Reproductive behavior, again, pretty telling, reproductive behavior and social interactive behavior. And this is relevant with the agriculture field, but also is an autism trait. And then the last one, resident intruder, help us check for aggression and social interactive behavior, which is also relevant for the agricultural field, but it's also an autism comorbidity and autism trait. And for today, I will be presenting some data on open field, elevated plus maze, and 3-chamber. So the first thing I want to go over is the elevated plus maze, which is helping us test for anxiety. And if you remember the expectations we have based on literature is that treatment animals are going to have more anxiety, and this is going to be shown more on males. However, with our data, again, pilots are good for the good, the bad, and the confusing. And this is confusing. So for our data, it's just that the treatment males show reduced anxiety-like behavior. The elevated plus maze is actually created with 2 open arms and 2 enclosed arms. So the arms have walls. Typical mouse behavior is that they don't like to be in open fields. So they're gonna reach for those closed arms, they're gonna reach for the corners. However, they're still going to investigate somehow. What we were expecting was that our treatment mice were actually going to spend less time in the open arms and more time in the closed arms, but that's not what we found out. So if you see here, I'm showing you the closed arm times in second, and then this is males, and this is females. Green is control, orange is treatment. And as you can see, our control males spend more time than any other group in the closed arms, but also they spend more time than the treatment males in the closed arms. Pretty graphs, looks really nice. Now, when you look at that, I wanted to check why is this happening. So I went a little bit more in depth in my data to see where each mice or each cohort was at. And I did this. So if you now see here, for males, the data look pretty bimodal. And this goes a little bit into rigor and reproducibility with your data. So just a little, please check your data point. So as you can see, the data looks pretty bimodal. I saw that, I thought it was normal. Nathaniel Sharp saw that, he said, that's not normal, please look into it. So when I look, well, this is not showing us that it should. Okay, well, why is it doing that? Well, my graphs lost their color. So what this was showcasing is actually the same data, but I broke it out into cohort. And if it was showing the colors as it should, you would see that all here was cohort number 4. And then cohort number 4 was also here, whereas all the other cohorts were at the end of the graph. So the data was my model because cohort number 4 was actually making it like that. Once I removed cohort number 4 to check if the data was still relevant and if the effects were still the same, the effects were still the same, sadly, but they were not as significant. But still, even without excluding cohort number 4, our treatment males show reduced anxiety behavior. Now, this one was supposed to show the time spent, well, it's showing the time spent in open arms, which is the regression of that. And these are males and females. I wish I could see the colors. But this here is our treatment males, treatment females, control males, control females. And what we found out is that our treatment males were spending more time in the open arms. Makes sense because they're spending less time in the closed arms. However, this data here is not bimodal with cohort 4. And this is where the confusing comes in because we haven't been able to explain this, but again, this is data from our pilot. Few things that we think is creating this is that when you run elevated plus maze, ideally you should run it from 10 to 15 minutes. We only ran it for 5 minutes because we were just trying to make sure that our test was correct. So that's something that we are going to change moving forward is running it for a longer time and hoping that we can actually create data that has been published before, because right now it's just the other way around as we would expect. Now, when we look into open field, which is helping us to check for motor ability and anxiety, we find that our males, overall, our males, control males, and treatment males show reduced anxiety-like behavior, and treatment males show greater reduction of anxiety. Again, not what we were expecting, but now that we also have the open field to look back, maybe polyice is actually just decreasing anxiety in our males. And here what I'm showing you is the percentage of time they spend in the center. So open field is divided, it's just literally a box, and it's divided into two zones. So the inner zone and the outer zone. Like I said, mice really like the corners and they like walls. So that's where we would expect them to be the most, but we would also expect them to investigate a little bit. So as you can see, our control males, they spend most of their time around the walls, but they did spend some time in that inner zone. The red is showcasing where they spend most of the time. The blue is where they spend less of the time. And as you can see, our control males behave as you would expect a typical mouse to behave. For our treatment males, they did the same thing. So they spend most of the time in the outer corners, and they did investigate at the center. The reason as to why you don't see that red dot is because our control male investigated at the center, but they stood in the center, whereas our treatment males investigated the center, but they just moved around the center. So it wasn't as like they felt comfortable in the center, they were just investigating the center. Now, when we look at our control females, we see that they spend most of the times in the corner and then some investigation in the center. Whereas for our treatment females, they really do not like the center at all. So they spend most of the time in the outer walls of the open field. And lastly, this is showcasing some data from the 3-chamber assay. And the 3-chamber is helping us test for social recognition. And what we found out is that males show greater social preference, and we really didn't see a treatment effect. So for right here, you're looking at two different graphs. This is stage 2, which is where we have our testing mice with stimulus mice in a little cage, in an empty cage on the other side of the apparatus. So here, we're looking at the social preference index. So I did a really cool math that is always done for the 3-chamber that gave each mice a social preference index. And what we found out when we did this is that our treatment females, we have more treatment females with negative social preference index compared to any other groups. And then when we move on and we look at stage 3, when now you have that familiar mouse, so the mouse that they saw before and they investigated a little bit, versus a novel mice, a new mice, we see that most of our groups, well, all of our groups now have half or even more than half of their mice with negative social index preference. And same thing for our treatment females. So lastly, after all that behavior work is done, we move on, well, I move on to do transcriptomic assays workflow, and we just track the brain, we dissect brain regions of interest, then we do RNA extraction and data analysis. The regions of the brain that we were interested on for stage 1 were the medial prefrontal cortex, which is associated with social interactions and anxiety, the medial preoptic area, which is associated with mating and parental behavior, and the ventrolateral subdivision of the ventromedial nucleus of the hypothalamus, the VMHBL, which is associated with aggression and mating behavior. Now, so all this data that I show you guys actually came out from the pilot. So the pilot, it's done. We have completed our N, we have completed all behavioral analysis, And now we're just waiting to get back our transgenic data sequences to be able to analyze that. So what's next for me is to analyze the genomic data from stage 1, because we really want to see if there's anything that is being changed or affected at the molecular level with MIA and sex differences. Another thing is that I want to rework my behavioral assays a little bit and select behavioral assays that can help us test the 4-core genotype mice. So I'm going to keep some of the ones I have done, but I also want to add additional ones that test for sensory abilities and other communication skills. And then lastly, do RNA-seq on additional brain regions. So add more brain regions that are directly connected to key behaviors or that are implicated in human autism. And then just for some topics, validating research resources, we ran pilot 1. This is helping us a lot to finalize what we're going to do with the 4-core genotype mice, which it's coming in December. Jack's had it locked for the past 2 years, and they finally opened, so they're coming in December. I think this is the first time the university is going to have them on ground, so that's pretty cool. And then blinding and masking to reduce expectation bias, because I run basically almost everything from setting the cages up all the way to behavioral assays and data analysis. Obviously, I shouldn't be told what treatment each mom got. So for the first cohort, because it was just me, I actually did do the injections and I ran the behavioral assays. But between that, we had Jules from the Weisman Center blind the mice. So he changed the numbers. I didn't know which mice I was running. Now, because I have Kevin and Matthew, they actually are the ones that are giving the injection, and then I go into wean. So by the time we go to do behavioral assays, all of us are blinded because I don't know which mice got the injections and they don't know which pups belong to which mom. And then figure and reproducibility, I spoke a little bit about that with our elevated plus MACE. And then considering the effects of biological variables, age, sex, weight, psychological states. It's not here, but I really want to talk about the hormonal stage of the females because we're running behavioral assays. One of the things that can affect the way they behave is their hormones levels at the time we're running behavioral assays, or our stage of the estrous cycles they're at. And the way right now we are considering for that is by just checking the estrous cycle. So I take acknowledgement of what stage of the estrous cycle they're at, and we use that as a covariate when analyzing the data. On top of that, the stimulus mice we're using are overoptimized females. So there are females who their ovaries have been removed. That way we know that their hormones are not affecting the way the males are behaving towards them. Yeah. And then lastly, I want to thank my committee, Donna, Nathaniel, Dr. Reed, Dr. Shinyu, and Dr. Wayati. Wayati left. If anyone wants to join my committee, please. And Jules, who has been a lot of help. And I also want to thank the people in my labs and the undergraduates. I take a lot of pride in mentoring undergraduates. So by far, I have had 8 of them. They all stay with me. They just have graduated or Phil has taken them. But specifically, I want to thank Kevin and Matthew because all those behavioral assays that were ran at night because of the dark cycle that mice prefer. So it was long 9 hours of 7 p.m. to 7 a.m. All 3 of us in the Weisman Center. So I want to say thank you to them and Matthew because he can get his PhD with me if he wanted to. So, yeah. So thank you. Yes, Myron.
That's super interesting. You mentioned at the end with the female hormonal state you're keeping track of that. Do you have any expectation of how that should bias the data one way or another, or have you looked at that?
Yeah. So not to be biased, but we all know that when females are on the estrous cycles, the hormonal stages are different, and the way they're going to behave towards other animals, especially males, is also going to be affected. So once they are in that active period where they're actually getting their cycle, we expect them to now want to, like, first of all, females mice, they don't really like to do a lot of male mice, but during that stage, we expect them to really not want to socialize with them at all. So that's why it's so important for us to make sure that we're taking into consideration their cycle, and also as much as we can, I try to make sure that we're running this those really strong social interactions, so the 3-chamber and the true social interaction, while they're not in their active cycle. Yes.
Moving off of that, you talked a lot about the males or the females and their hormonal how it affects their males, and especially living within males being close to females, and if that's changing how their hormones being maybe interacting with the social behavior.
Yes, so data have shown that the males don't really get affected a lot by their own hormones, but their behavior can get affected a lot by the female hormones. That's why we use those overactinized stimulus mice. We take the ovaries out. It's not as bad. I've done the surgeries. They are good to go. So we take their ovaries out to make sure that the stimulus mice we're having are not going through any hormonal cycle, but also the animals that our treatment animals, our control animals, they're caged with their same sex. So at day 18, at postnatal day 18, they get weaned. So basically they get taken away from mom because they can't survive on their own. They're caged with same-sex animals. That's why it's also so important for us when we do the 4-core genotype to know which group they belong to, because instead of just having XX with ovaries and XY with ovaries, so 2 cages, we're gonna have 4 different cages. Another thing we do is that if a male is a single cage, we don't use them for behavior, because that can actually affect a lot how they behave. Yeah.
You say that you're looking at just males are diagnosed with autism at a lot higher rate than females, correct?
Yes.
But I've also read suggestions that the reason is because all the autism diagnosis, like a lot of other things, are geared towards just male and female presentation and not being better.
Yes.
How does your
Yeah. So the question was that when we look into the sex disparity with autism diagnosis, one of the reasons that has been discussed is that what is used to diagnose for autism actually focuses a lot in male presenting behavior and not a lot of female presenting behavior. And that is something that has been discussed. It's also something that has been, that the people that do clinical work are trying to change, because one of the biggest issue that they believe is that females are pretty good at masking their behavior. That's why a lot of females don't get diagnosed until later on in life, when they figure out that there's something that might not be that is not right with them, and then they go in to check. As for us, there's not much we can do, because we cannot diagnose mice with autism. We can only say they're presenting autistic-like behavior. So that's one of the ways we cannot. I cannot tell you my mouse is autistic, because I can just follow data. But also, when we are breaking down chromosomal effects from gonadal effects, is really going to help us see what is causing this big disparity. Is it the chromosome? Is it the Y chromosome? Or is it the hormones that the testes are creating? Or is it the hormones that the ovaries is creating? Is there some type of protection based on your chromosomal makeup? So that's the most we can do in our side. Changing the criteria for being diagnosed is more on the clinical side, which I do not know. Don't do that yet. Hopefully soon. Yes.
Following up on that, if you find no significant effects, are you then going to make the argument that the effect in humans is largely diagnostic based, or?
I cannot make that argument, because I cannot diagnose my mice with autism. That's the way I can put it to you based on the data. However, I am a huge believer that there is a big bias with the diagnostic criteria. in autism. So I believe that once that's fixed, that ratio is gonna go down a lot more. But that is not, I cannot make that argument because, again, I cannot diagnose mice with autism. If I was to do this in humans, and I was able to find data like that in humans, then that argument can be made with the diagnostic criteria. Yes.
The levels of the poly-IC affect the anxiety level?
Well, yes, but it's not that it affects the anxiety level, it affects at the rate at mom's immune system gets activated. So we use 20 milligrams per kilogram of the mom. And that is what has been used to do work like ours. If we wanted to see more, if we wanted to see behavior that has been more altered, we would either increase the poly-IC so mom's immune system is working more or working overtime, or we wouldn't even use poly-IC, we would use the actual virus, so the influenza virus or something else like that. Because poly-IC, technically, it does not make pregnant dads sick. It just activates their behavior. If we wanted to check for something that's gonna change the offspring's behavior completely and affect them completely, we would introduce an actual virus like influenza, which has been done and has shown different effects, but it has done more of like what antiviral medicine can we introduce rather than how is it actually affecting at what level it's affecting.